So I would like to ask Agnes Kant, who will chair the next session. Agnes Kant is director of LAREB. We all know her, I think, very well, many times on television and in the news during the COVID and other safety issues. So thank you for joining us, Agnes, and give the word to you.
Thank you, Miriam. So we're now going to go to the session on observed expected analysis and use in the safety studies. And at first I want to introduce the first speaker to you, which is Gordillo Maranon. There she is. And she's a healthcare data science at the EMA, and she's supporting activities in relation to the COVID-19 pandemic and performing data analysis of the real-world data and support regularly decision makers. So the floor is yours. She will give us a presentation about the methods that are applied to spontaneous reporting data and the use of background rights.
Thanks. So I'd like to thank the organizers for the opportunity to present today on the methods applied to spontaneous reporting data and use of background incidence rates to support the safety monitoring of COVID-19 vaccines. And in particular, today I'll focus on the observed expected analysis, how they were performed at EMA. I will also illustrate it with two case studies, and I will wrap up with some lessons learned throughout this period. So the disclaimer slide that Catherine introduced before. So I'd like to begin by providing some context about the COVID-19 vaccination in the European Union. So since the vaccines were approved, they have been extensively used in the EU, with over 981 million doses administered as of the end of last week. So this high vaccine uptake led to an unprecedented volume of safety data from spontaneous reports, with over 1.8 million adverse reactions originated in the EEA and reported to Eurovigilance, which is the system to collect, manage, and analyze adverse reactions to medicines authorized in the EEA. We can clearly see in this graph how the number of case reports increased since the vaccines were approved in December 2020. We can see in the pink, light pink, the safety reports related to COVID-19 vaccines, and how also the reports related to other products, they continue the same trend. So due to the nature of the COVID-19 pandemic and also this high vaccine uptake, the reporting of as expected adverse reactions had been anticipated to be very high and much higher than for the H1N1 pandemic vaccines. And indeed, this prediction was confirmed because within 3 months of the deployment of the COVID-19 vaccines, the number of adverse reactions reported to Eurovigilance was 20 times higher than those for the H1N1 vaccines within the same time frame. And also, as you can see in the graph, whereas the H1N1 pandemic vaccines reached a plateau after a few months, the reporting levels for COVID-19 vaccines continue to grow steadily. Another challenge for the COVID-19 vaccines was there were no pre-pandemic or mock-up vaccines, so this led to a fast development of vaccines authorized following the rolling review, which is a regulatory tool used by EMA to speed up the assessment of promising medicines under public health emergencies. So this whole experience led to numerous lessons learned on methods, data sources, and collaborative work, and I will present some of this in the next slides. So as I mentioned, the high volume of safety data had been anticipated prior to the rollout of the vaccines, and EMA put in place some specific activities to ensure the enhanced monitoring of emerging safety information. And this set of activities here called the monitoring toolkit combined routine signal detection methods, ad hoc statistical methodologies, and real-world evidence from observational studies. So in terms of routine signal detection in Eurovigilance, this has a component of disproportionality methods, which such as the reporting of ratio, and this was enhanced by increasing the frequency of the production of reports, and also by the development of a list of vaccine-targeted medical events, which was largely based on a list proposed by the Brighton collaboration under the safety platform for emergency vaccines. In terms of observational studies, well, Catherine covered that in the previous presentation, but EMA commissioned independent observational studies to generate background incidence rates for adverse events of special interest, to monitor special populations, and also to assess the association between vaccination and safety outcomes. And EMA also used ad hoc statistical methodologies, such as the reporting rates, to investigate the impact of regulatory actions and also differences within vaccines. And we also use the observed to expected analysis to compare the number of observed cases of an event following vaccination and reported to eurovigilance to the number of cases that would have naturally occurred in the same population without vaccination, the so-called expected cases, and are estimated for background incidence rates. So I'll focus on the next slides. I go into the observed to expected analysis and how they were instrumental for contextualizing and characterizing some risk related to the COVID-19 vaccines. So to perform an observed to expected analysis, one needs 3 key elements. So the observed number of cases, the risk period or the time period for which there is a suspicion or biological plausibility that there is a vaccine-associated increased risk of experiencing an event, and the expected number of cases. So at EMA, the analysis we performed, we used the observed cases from spontaneous reports in eurovigilance that were originated in the EEA. The risk periods were generally determined based on published information, biological plausibility, or taking into account the time to onset distribution. And the expected cases were estimated using vaccine exposure data from the European Centre for Disease Prevention and Control, combined with background incidence rates for adverse events of special interest generated by the Access Project or ascertained from healthcare databases to which EMA has access to. We also perform sensitivity analysis using different background rates, risk periods, or case definitions to look into more broad or narrow definitions of the event. So the first use case I wanted to present today is about the thrombosis with thrombocytopenia syndrome and the VAC4EU vaccine. So as you may recall, at the beginning of March in 2021, the Austrian National Competent Authority alerted the EUR regulatory network of unusual cases of thromboembolic events following administration of VAC4EU. So a few days later, the EMA's Pharmacovigilance Risk Assessment Committee initiated the evaluation of thromboembolic events, and this led to the identification of a new risk for a rare combination of thrombosis with thrombocytopenia. So the evaluation of this signal was very challenging at the time because when it emerged, there was no case definition, no background incidence rates, or specific measure attempts. So the early OE analysis used proxy background rates to investigate the safety consent, and this included coagulation disorders, disseminated intravascular coagulation, and cerebral venous sinus thrombosis. And here we can see the results for CVST. We have a forest plot on the right, and highlighted in red, we see that the number of observed cases was higher than expected and statistically significantly higher, especially in the younger age groups. So this information was included in the assessment report, and by the 24th of March 2021, the product information for VAC4EU was updated, and healthcare professionals and the public were warned about the risk. The second use case I wanted to present is myocarditis and pericarditis and the mRNA vaccines. And in this case, the starting point was different because myocarditis and pericarditis had been associated to the SARS-CoV-2 infection and therefore were included in the list of adverse events of special interest. So just to give you some context about the signal, this emerged in Israel for Comirnaty in early 2020, where most cases that were reported were mild and predominantly in young male vaccinated after receiving the second dose of Comirnaty. So this triggered a review of myocarditis and pericarditis at EU level, and 145 cases of myocarditis were observed in Eurovigilance that originated in the EEA countries for Comirnaty. So then Prague requested OE analysis for this specific safety concern. And here we see the results stratified by sex, so males left, females on the right. We also, it's also stratified by age group and by vaccine brand. So at the top you see Comirnaty and at the bottom is Spikevax. And again, the statistically significant OE ratios are highlighted in red, and they show how the observed were higher than expected for the young male groups. So this information again was included in the assessment report, evaluated, and by the 9th of July 2021, the product information for these two vaccines was updated and the risks communicated to healthcare professionals and the public. So based on our experience from implementing and interpreting this OE analysis, we have learned very valuable lessons on facing a massive influx of spontaneous reports, on comparability between observed and expected analysis, on dealing with incomplete data, and on generating, selecting, and using background incidence rates. So I will give you some lessons within each group. So first, we learned that OE analysis provide an additional tool for the safety monitoring of COVID-19 vaccines, and it provides a one-to-one comparison, which is independent across vaccines, and this is different to what other statistical tools does, because the reporting of ratio, for instance, it compares with other medicinal products in the database. We also noted that one of the main limitations of observed to expected analysis is the underestimation of observed cases that may occur due to the underreporting of some events or the delayed reporting. We also learned a poor alignment between the codes used to identify the observed cases in the spontaneous reports and thus to calculate the incidence rates and get and estimate the expected cases may affect the robustness and interpretability of the OE results. Third, the missing data and incomplete data is a challenge, and we had to make assumptions and imputations to deal with it. And lastly, on background incidence rates, well, they were ascertained from national databases, which may not be fully representative of the EEA. And we also noted, as many others, that substantial heterogeneity exists across incidence rates between databases and or time periods. So just a few key messages to conclude my presentation. I think we all agree that the pandemic has been a huge booster for the development of tools and methods to deal with the unprecedented volume of safety data, and also to improve evidence generation based on secondary use of data. From the regulatory perspective, the need for flexibility and efficiency, as well as the value of scientific collaborations, have also been important lessons learned. In terms of OE analysis, there are several sources available of background rates that help addressing emerging safety concerns, but these need to be regularly updated for adequate preparedness for future health emergencies. So it's the lessons learned that have been presented by Catherine that we'll discuss today are important to inform and support preparedness for potential future health emergencies. So with this, I'd like to thank you for your attention. I'm happy to take any questions.
Thank you very much. Maybe some questions from the audience. No one? I have one question. Oh, there's one question.
Yes, Vincent Bauchau. So this is a very good example that observed to expected analysis can indeed confirm a signal when there is something. Now, by looking at your observed to expected ratios across the gender and the age, it looks very similar to what the studies have produced. And have you tried to compare the observed to expected ratio to the relative risk from the studies?
So we haven't, well, thanks for the comment because it's, we definitely will look into it. And the observed expected analysis actually were produced before the studies. And we hadn't compared them in detail how they perform. Yeah.
I don't want to be very provocative, but the observed to expected, you can do this in a very short time. Studies takes more time, and this, and again, it's very provocative. Do you need to do any more studies? They come very much later and confirm. It's very provocative.
Yeah. Well, OE, yeah.
Yes, go.
Well, the observed expected has many assumptions and also challenges. So there may be other methodologies more robust to investigate, but they have proven very key at the earliest stages of looking into a signal to generate some evidence and guidance for next steps.
Thank you.
Thank you very much. We're going to get the discussion in the panel, so a lot of issues will come by. I have one short question. Did you look also per country? Did you do a stratification per country? Because the reporting rates per country are very different, and then you could look maybe to the difference between the countries.
Yes, so towards the end, where vaccine coverage was not provided for the whole EEA, so some countries stopped providing stratified vaccine exposure data, then we did look into per country OE, but then you lose the power of your number of the cases. But yeah, that's something we look at.
I would like to see them, but that's for later. So we go to the next speaker. Thank you very much. Well, the next speaker you all know, Miriam. You're on yourself. Or the present. Well, give the present. Okay, I will give you the present. Yeah. So you all know Miriam, so she doesn't need an introduction, but I will do anyway, because she's a professor in real-world evidence and head of the Department Data Science and Biostatistics at University Medical Center in Utrecht, but also the coordinator of the project Early COVID Monitoring, where we are talking about today, and or the co-founder and president of VAC4EU. Miriam, go ahead.
Well, thank you very much. We now shift to a method that's not based on spontaneous reporting anymore, but was actually done as part of the early COVID vaccine monitoring, where there were two pieces, actually, the cohort event monitoring, the inclusion of vaccinees, which we'll talk about later, but also monitoring of electronic health record data. So we tried to be fast. A couple of weeks or 1 month seems very fast. If you can pull up my slides, it would be great, yeah. So let me talk in between. So what we tried to do was a near real-time monitoring, which was based on lessons or proof of concept we had done actually in the advanced study, where we created this beautiful dashboard and did this pertussis retrospective proof of concept. But now we were dealing with a real-world situation, a pandemic, and we tried to have this dashboard where you could actually monitor the incidence rates post-vaccination and compare them with the background rates. Good. So I go back now. Yes. First of all, before I go, I would like to thank all the co-authors and a tremendous group of people that have been participating in all of this work from various organizations. So this project that was quick actually after the vaccines were rolled out was really to provide or to build knowledge about product safety as soon as vaccines came available. It was not testing hypotheses, it was just monitoring. So I was just saying, today we are going to focus on this method. We will hear later about the cohort event monitoring, but this was monitoring electronic health record data. And of course, they need to have short lag times to participate, not need to wait a year, because otherwise it's not very informative. And we built an interactive dashboard for EMA to actually show what the rates were and reported on interim in July and then in December. So the design of this study, very briefly, there was a 2020 was utilized as the background rates. Then there were non-vaccinated after the vaccinations were rolled out, but also those that were vaccinated. Their person time was split in weekly intervals, and for the main analysis, it was after each dose 28 days, unless you went to the next dose. The events were accumulated actually, so to calculate the post-vaccination rates and compare that with the prior, with the rates in 2020. We chose 2020 because this was the time of the lockdown where healthcare access was less than usual. So the choice could also be to use rates from 2017 or 2019 that we had estimated in access, but we wanted to use this because of lockdown measures. All of the rates were done, differences were calculated and also adjusted rate ratios. And then meta-analysis with random effects. So these were the data sources that were available and could actually participate with having relatively short lag times. The Pharmo database, they kept for this analysis only the GP data because they have this short time. If they link it to hospital, they need to wait for a long time, so the GP data. The Spanish data from the Spanish Medicines Agency, the BFAP data source, the primary care, but also linked to hospital data for part of the regions. The Tuscany Regional Health data source and record linkage data source, and then the CPRD data that was handled by the Utrecht University. And with different types of outcome assessments, both hospital-based from BFAP and ours, as well as GP only. These were the events that were studied, a whole list, actually all the ASI plus transverse myelitis and Bell's palsy, which had been added because some concerns had arisen in trials that were ongoing or signals after access. So the way this was worked, we used a common data model. So data sources were local. They transformed their data, they ETL'd their data into the conception common data model, conducted quality checks, then ran the script for semantic harmonization and all this creation of the study variables. Then the analysis was done also using our scripts. The aggregated results were sent to a remote research environment, Andrea. There they were pulled and put in a dashboard by the team in Power BI and then made available. So here, shortly, the study population. This is from the final analysis. So you see the largest is CPRD in the UK up to May 2021. Then the Italian, the Tuscany data, slightly longer until July. The data from BFAP here, the primary care, and then for a small piece also those linked to hospital data. Now, a couple of results that many of the ASI were not significantly associated with any of the vaccines. So I'm showing very quickly, it's not about the results today, but what was picked up. Anaphylaxis following Vaxzevria, actually. So here you see the database specific estimates and the pooled estimates. Then erythema multiforme after Moderna vaccination. GBS after J&J, Janssen vaccine, also something we know and has been discussed in Prague. Venous thromboembolism, also after actually Pfizer and Moderna, and we didn't see it at that moment for the AstraZeneca, but it was associated. And then TTS, the proxy that the case definition that was utilized with having any arterial or venous thromboembolism together with thrombocytopenia within 10 days. And what you see here is the significant association that was picked up for AZ and also J&J. So the study design was able to pick up those signals actually that we are now all aware of. There were also other observed expected analyses. This was not the only one, of course, done. The Danish and the Norwegians published a separate study where they looked at the period from February 9 till March 2021 to only those vaccinated with Vaxzevria. And compared that with their background rates in their country. So also using the same data source to see the observed as well as the expected. And the main outcome was also 28-day, and they looked at hospital contacts, and they noted that among recipients of the ChAdOx vaccine, increased rates of venous thromboembolic events, including CVST, were observed. So also higher observed than expected. So this is just another way to also do observed expected. Instead of observed from spontaneous report, use the same populations to do observed expected. And I think we need to discuss what place that has in comparison to the observed expected that we just have from spontaneous report. Also, the Erasmus group, on top of, I think Catherine pointed out, they were doing a study on the thromboembolic events and topped this onto the added the vaccine component using IQVIA data sources, CDAP and CPRD, as well as IPSI. Data were reported only for CPRD and CDAP. So for CDAP, and they were actually reported separately. So for CDAP, they compared the rates of all thromboembolic events and TTS, and findings suggested that actually there was no higher observed than expected in the CDAP database for these events. There was also analysis done in CPRD, and here is the picture of that, looking at blue is for Pfizer vaccine, first dose, green, second dose, orange, red is first dose for Vaxzevria, second dose Vaxzevria, and then the orange here is COVID cases. And you see here that there is an increased SIR, the standardized incidence ratio for cerebral venous sinus thrombosis. After the vaccine, also immune thrombocytopenia, and there were no other significant. You see the orange one, which is these rates after COVID itself also are increased. So COVID itself also increases these events, which is important if we do evaluation studies, because you need to control for that. All right, conclusions are different groups conducted observed expected analysis within same data source. Background rate period differed, so we may want to think or discuss whether, what would be preferable, but this may cause differences. So either further in the past or 2020, that was used in the ECVM. Post-vaccination calendar period differed slightly. We need to see also, we haven't thought about that carefully, what impact that may have. So in the Nordic collaboration, they used that period that prior to the signal on AstraZeneca vaccine, whereas the other studies used all of the calendar period that was available at that time. So you actually are mixing an effect potentially of prior to the signal and afterwards, where there were actually risk minimization measures already in place. So it's actually interesting to see whether that has an impact. The reporting patterns differed. So many of them did actually a meta-analysis of the data. So the Danish and EUP, but the other Erasmus was actually separately reported. And adjustments differ. So most of them only age and gender, but we saw initially when we were rolling out that there was strong confounding, so we also adjusted for risk factors that were associated with severe COVID-19, which impacted quite a lot. Thank you.
Thank you, Miriam. Questions from the audience? Yes. Maybe Xavier first, and then behind you. Yes, please.
Yeah,
go ahead.
Just a comment on the Nordic evaluation of the AstraZeneca signal in relation to observed versus expected and potential methods issues was that in Denmark and Norway it was used in frontline personnel, right? So there was an obvious potential for confounding when you use an observed versus expected method. The point was just that we were fortunate that the signal was so strong that we picked it up easily with the observed versus expected. But for other events, it would have been something to address in further evaluation studies, I would think, yeah.
Okay, thank you very much on this. Any other question? Yes, Xavier. behind.
Yes, thank you. Miriam, in the review of all the studies and methods that you made, have you identified any differences in results, not numerically, but overall, that could lead to different conclusions about the risks? It seems to me that there has been several observed to expected analyses, several studies, but all finally came to the same conclusion about the difference in the risk between vaccine, between age group, and so on. Okay. So the question is, we can of course discuss the differences in results in the quantitative results, but the question is whether there have been different, could it lead to different conclusions and from a regulatory point of view?
Yes, I think so. So without having it on the top of my mind now, but for example, when this was rolling out, the first reports, I think especially on CDAP and CPRD showed no increased risks actually. So later on with additional data, they were coming out. So whereas in ECVM, for example, initially we saw very high risks for death, which were related just on the very strong channeling. So you noted that doing this like recurring, or others call it rapid cycle analysis potentially, you risk a lot in the beginning because there was such a strong channeling actually of the vaccines that you could have taken wrong conclusions. So some time needs to pass to be able to accumulate the events. I think that is really important and you need to adjust. And I think drawing conclusions too I've also noted is that there is a lot of effect modification by age and gender, and that all of these vaccines have very strong age-gender related patterns. So even this like observed expected analysis like this, I mean, you need to stratify for age basically, otherwise you would have missed signals. So I think you noticed this for AstraZeneca because it was used for healthcare professionals then, Anders, I was not aware, but that's good. But for example, in the ECVM, we didn't see myocarditis yet because we just did like a total population. And so you can also miss things. So we need to explore that better.
Yeah, thank you. Discussion already started. Thank you. So we're now going to have a panel discussion. Yes? Sorry, I forgot. Yes, we're now going to have a panel discussion. I want to ask the panelists to come forward. And this is Vincent Bauchau. I pronounced it wrongly, I guess. There you are. Did I did it wrong? Bauchau in French. I'm sorry. And you are a co-founder of VAC4EU and a former head of pharmacovigilance of JSK Biologicals. And we have also already Xavier Kurz, former head of data analytics of the EMA during the COVID pandemic. And the last person is Florence van Hunsel, and she is head of innovation of signal detection at the Pharmacovigilance Center LAREB here in the Netherlands. And we're going to have some discussion about, I'm going to sit in the middle, I guess, yes? Oh, you have a watch, okay. We have 20 minutes. So we're going to have some short discussion about the observed expected analysis in the safety surveillance. And I want to start with the questions, what do you believe was the usefulness of this this kind of observed expected analysis in the different ways, in the spontaneous reporting and in the way it's what's used in the rapid safety analysis. And I want to start on the left to me, because that's the first in row in spontaneous reports, are always the first to see potential signals. So Florence, could you say in what way it was useful to use observed expected combination with spontaneous reporting?
Yes, thank you. I think a lot has been said already in the EMA presentation, but if I look at how we used it at a national center, how we use this method is normally spontaneous reporting, and the way we find signals is a very clinical way of looking at data. And then in our toolkit, we also have disproportionality analysis, so things we can do to find signals. But during the pandemic, we had such an enormous influx of data that just looking in a clinical manner at cases, you cannot take the background incidents into account. You need to find ways to, tell you something on this influx of data and what you are seeing. And for me, the observed over expected method, it doesn't give you a definite answer. It doesn't give you a definite answer on causality, but I do think if you use this method, especially if you use it in a stratified way where you look, where you take into account age and gender, like has been said, it gives you a better idea if there is a potential signal there. And of course, you have to take into account mechanism, all sorts of other data, but it does give you a better grasp on what you are receiving. And I think that is the way we use this method, at least at our center in the Netherlands.
Mm-hmm. Yes, and we saw the signal, I know because I was there, we saw the signal on thrombosis in April 2021 already in observed expected analysis on reporting. But it's just to give the signal some more background. That's what you say, okay. What do you think, Xavier, what is the usefulness of this kind of analysis for signal detection?
I would like first to stress that the observed to expected analysis is not a weak method. No. The method itself is rather strong. It is based on the SMR. There is a strong theoretical foundations. Now the problems when we start using it with in this context is the uncertainties on the numerator and denominators. And this is not really a matter of random variability because this can be handled with the confidence intervals, but this is more as a matter of bias and perhaps lack of comparability between what represents the numerator and denominator. And that's why at EMA we did a lot of work, as explained by Maria, to try to identify which were, first of all, the observed cases that would best would apply, what would be the typical cases and typical patients, but also for the expected number of cases, what were the data that we have who could be valid enough to represent the observed cases. And that's why there was a need for a lot of assumptions. But overall, I think that all the signals that were detected through observed to expected analysis and put in the SMPC have not been refuted by further analysis. So I think that this is perhaps show that when handled correctly, I think this can provide valid result, even if there are a number of uncertainties that need to be handled.
It proved to be useful.
Of course.
Yes, yes. Vincent, what do you think?
Yes, I had a question, but Xavier just answered the question, but I will repeat the question again after. But Xavier is right, there is a strong methodology but a lot of uncertainty, and there's a sort of art of doing the proper observed to expected analysis. And for example, if you use the wrong time to onset, you can easily miss the signal. I haven't done any observed to expected on COVID vaccines, but I was doing observed to expected on the interception after rotavirus vaccines, and it's only when one day we realized that the time to onset was probably only the first week, and also we said we should separate the first dose, and at the time the sample size of cases was sufficient, and then suddenly there it was. While you can easily miss, if you take 14 days, you will miss the signal. But the point about observed to expected, you can do it in days or even 1 day. I agree, sometimes the background incidence rate is missing, and then we can go back to background incidence rate. The good thing about background incidence rate in preparedness is that you could produce them before the vaccine is used, and sometimes they also published before the vaccine is used. And for H1N1 there was this wonderful publication where the observed cases for 100,000 vaccinated cases were published, which then set the expectation. Miriam said there was, for example, an increase of deaths, but if there will always be cases of death after vaccination, but you can actually produce expected cases per age for so many people vaccinated. So it's very useful method. Now, my question was how many of the signals how many decisions have been based on observed to expected, and you said that at least when there was a decision based on observed to expected, that was the good decision, which I think it's in terms of lessons learned, that is critical. That's a sort of lessons or message we should hear after all of this COVID crisis.
Yes. So it was useful. I already concluded that, but then there probably will be improvements possible. If we're gonna be, what are the lessons learned for the methods? Are there things we should do better in the future to use these methods in the right way, Xavier?
For me, I think that what we would need to improve is to improve the efficiency of generating background rates. We lost a lot of time in looking at the different background rate that we have, trying to identify those that were valid, those that we could use or not use, and probably it's because we didn't have enough information on what were the characteristics of the database, what was the patient population, and so on. And then it was very difficult at some stage to take a decision on what were which of the background rate we should use. And in terms of efficiency, there is also the issue that we met that about the mapping between the MEDRA terms reported in pharmacovigilance and then the terminology used in the databases, which were not fully compliant, and therefore Miriam know that we had to ask, do we do additional analysis, because we realized that the algorithms that was used for some areas were not fully compliant with the MEDRA term. So I think this should be an important improvement. I know that there is a mapping being done now between MEDRA and SNOMED, but this should probably be generalized. Yeah.
Okay. For the use in spontaneous reporting, Florence, what improvement should be made to do better?
I think for us as a national center, we were already quite lucky that, of course, there was the access background rate, but we were able to work with Pharma, Ron is here in the front, and have additional background rates for the Netherlands, which were very precise and specific for us. And we also had a linkage with the Dutch vaccination registry, which gave us, the best correct data that was available on vaccination rates in strata per age and sex. And I think that is really something that you need to do this correctly. And then I think what could be improvements is really think about the sensitivity analysis that was also already mentioned in the EMA presentation, because the reporting pattern can also be very different between types of signals. So maybe you have a lot of underreporting for a certain signal, but also stimulated reporting during media attention. So I really think that we need to think about how to do these sensitivity analyses to do this correctly and draw the right conclusions.
Yeah, thank you. Vincent, for you the same question. What do you think should be done to improve?
Yeah, I can see a few challenges. So first, the access and other groups have produced a huge volume of background incidence rate, and this is the first time where so much background incidence rates were produced. But it has a sort of side effect that then there was too much information. Sometimes you don't have enough information. Sometimes you have too much in the sense that then if you were looking for a background incidence rate, you could see all sorts of values depending on the database, the country, the period. And then indeed sensitivity analysis is there to help you. But and this is based on my experience as leading a task force among all the manufacturers. But they was at times confusing for people in pharmacovigilance to understand which background incidence rate they were using, and sensitivity analysis were, like thousands of sensitivity for a single observed to expected. You have to do a few, but then it was overwhelming somehow. And the other point is that the access and other background incidence rate derived from electronic healthcare records were great, but then there was this idea that came up that this was a gold standard, and people were even not willing any longer to use lit review to find background incidence rate, which I think sometimes you have pretty good background incidence rate in lit review, and at least you could have maybe some values for your sensitivity analysis. So I think we need to be careful. Too much information or relying only on one source of information. I think this is all what I call finding, or maybe describing or agreeing on the best practices for observed to expected to make it, state of the art observed to expected in the end. And it's that's a strong methodology, but there's a lot of, customization thinking to make it indeed able to detect the signal. I'm glad to hear that when there was something detected, it made a decision. It led to a decision which was not changed after. So this is fantastic, actually.
Okay, maybe the audience has some questions. Rosa.
So my it's more a comment is about, I think what you said, Xavier, is that the threat to, probably the biggest threat to the observed to expected analysis is differential misclassification, and the risk is of course higher when the source for observed and the source for expected are different, so that the differential is expected. But I think that one of the things that we could do in a readiness phase is to really perform systematic validation of our background rate, both looking for false positives and looking for false negatives, which is something that for which there is methodology development.
Thank you. Reactions on that? That was a comment. Saad, glad you're here. Always a good question or a good remark. Please, go ahead.
Thank you. Well, a key point. First, in the four main events, which we have the vaccines, thrombosis of thrombocytopenia, myocarditis, pericarditis, Guillain-Barré syndrome, and anaphylaxis. The myocarditis and pericarditis and Guillain-Barré syndrome and anaphylaxis, the background incidence and so on, can be found in, historically and be documented before the agent becomes available. That's the future. With regard to the thrombosis and thrombocytopenia, because it was almost a unique syndrome similar to the heparin one, it has special characteristics: thrombosis, thrombocytopenia, high fibrinogen, the P antigen, and so on. So the expected was central venous thrombosis, which is not quite the clinical condition which became after the vaccine. It didn't matter because even central venous thrombosis, taking the age consideration at the time, was rare, but actually the whole syndrome was very, very rare. It didn't matter. So that, we can't have that before. That we would need to develop and find the expected quickly because of its very, very rare characteristics. The second point which I want to raise is when the background incidence is very rare as opposed to sort of more common. When it came to anaphylaxis, which is common, when the observed to expected becomes more difficult to ascertain because the background is higher than very rare. So it will need kind of more careful and more robust epidemiological evaluation with numerator and denominators to try to validate the data from spontaneous reportings. So what I'm saying is some events we can have the background incidence, others we didn't, and we used the surrogate, which is central venous thrombosis. And the other one, what do people think about when the event is relatively common, like anaphylaxis?
Yes, good remarks, Saad. So it is useful, but it is very different in the usefulness depending on the signal, of course. Because what you said of the TTS, maybe Florence, you could get into that, because that's discovered because of spontaneous reports, because of very specific side effect. Maybe you could say something on that.
I think for us at a national center, actually we didn't even need an observed over expected for that signal because it was triggered by literature. You have such a unique event. You have people working on the mechanism right away, and the number of data coming in was relatively small, so it was much more a clinical exercise of looking at the cases, what we see here, than using it observed over expected.
Yes, but it's quite different if you just looked at the normal venous thrombosis. Then you have a totally different situation, and then it could be useful, observed, expected, because we saw also more observed than expected in that one. Other remarks? Miriam?
Yes.
There's the microphone.
So just a question, because I think for the ones that are ASI, we have methods in place, and then we have this weird, you can call it ASI-X, you know, the one that we didn't know was ever associated with vaccines, and we saw it in H1N1 with narcolepsy, now we saw it with TTS. So what do we need to have in place to discover this ASI-X? Is it just a clinical network that is, we just need to make physicians aware that if they see something out of the odd, that they report it immediately, and or you know, signal. What do you think?
I think, well, I'm it's not supposed to answer the question, but she will.
No, but we agree. No, I think finding that signal, you really need clinicians, and you need clinicians to be aware, you need clinicians to report, you need them to work on a potential mechanism, and I do think for this particular signal, clinicians work together throughout Europe to really quickly gather information on this particular unknown event. And I think that is the best way in finding the signal, and then later on, you can do observed over expected, but first you need the clinicians and them working together and reporting. Otherwise, you won't find it.
Yeah, thank you. Gianluca.
Yeah, I was thinking that observed versus expected analysis is one of the rare examples in which we combine together data from spontaneous reporting databases and electronic healthcare databases. And the feeling I have is that people working on one of these sources is not so much familiar in the potential limitation of the other source. And I think this may be a challenge and a limitation in making benefit of all the data sources we can access for vaccine safety surveillance. I don't know, what do you think about that?
Yeah, I totally agree with you. So I think that is one of the things that we could do even better. Hopefully a new pandemic is a bit away from us, but that is something we can do better.
Yeah, and not only during pandemic, regularly. Xavier, Vincent, do you have some remarks to close it, the discussion? Your last remarks, please.
No, just I had a remark regarding the question that Miriam asked. I think one of the challenges of pharmacovigilance, and you know it better than me, is to identify the 2 or 3 cases of the first case of TTS that are reported among 1,000 cases of diarrhea and other things. So I think the challenge here is to have the system in place at the level of the regulatory authorities and agencies to identify these rare cases and not to pass over, say that it's only 2 or 3.
Yes, but then get the discussion back to the more normal events like thrombosis, normal thrombosis. Then it's difficult because we had thousands of reports on that also. And then it is the normal way this disease goes, and then you don't know. So then you need other methods. Because spontaneous reporting does not give you the answer. So then you need another way.
And I think to me that's a very critical discussion. It's a bit away of observed to expected than the rest, but so it seems like most of the signals, important signals, were detected by clinicians, and not necessarily at an agency, but seeing an unusual increase in number of unusual events. Now that's fine, and they are probably the better signal detection system in place, which I tend to call the lonely country physician maybe. But then, because narcolepsy, that was the case. They were sleep specialists detecting an unusual number of cases in their practice. Instead of 2 per year, there are 10 per year or whatever. But then there was a delay before they were communicating this to regulators, and one of the delay was because they wanted to have publications, and there was this wrong idea that if you submit something to the regulatory authorities, then you can no longer publish. No, that's a sort of, if we truly believe that clinicians have specific rules, and they do, they should be maybe at the start of any pandemic a message clear to all the clinicians. If you see something, please report. And by the way, maybe editors of journals would reassure them, generally all clinicians, don't worry, that will not prevent you publication, which is a need and for the clinicians. So, sorry, it's a bit away from observe to expect.
A little bit.
But lessons learned from the COVID are very critical. It's a unique situation, and I think we can learn a lot. And in terms of the future, I have another project looking at the future of what we can sustain from COVID and how to maybe look at how we should put our resources in the future. But for signal detection, I think we should really think where we have to put our resources and facilitate some of the existing systems which are maybe more efficient and critical. And system is a very large word, which is a data source and a sort of process, general process.
Thank you. I want to stop here the discussion. Thank you all. I think the conclusion is it was useful using this, also observes expected, additional to the other things that are already in place. Thank you.